Let's look at cell division meiosis 2. So after a cell has completed meiosis 1, which results in 2 cells, it will proceed to meiosis 2. So meiosis 2 comes after a typically brief of interphase in which no DNA synthesis occurs. Then the second meiotic division begins, and this meiosis 2 resembles the normal mitotic division. So all the stages which takes place under mitotic division is similar to meiosis 2. So meiosis 2 involves 4 stages or 4 phases, which are Prophase 2, Interphase 2, Anaphase 2, and Telophase 2. So let's see what happens in all these phases. Remember, we are going all through these phases with 2 cells which have resulted during meiosis 1. So under Prophase 2 of meiosis 2, what happens is that the nuclear membrane breaks down and the new spindle fibers are formed, and then from there the centrosomes appears. The new centrosomes appears. So these are the 3 processes which takes place under Prophase 2. And then from there, after Prophase 2 has completed, then the cell moves to Metaphase 2. So during Metaphase 2, the spindle fibers which have been formed under Prophase 2, they will bind to both sides of the centromeres of the chromosomes, and then from there the chromosomes will align at the metaphase plate. Okay, so you can see here we have these chromosomes. Remember, these 2 cells result from meiosis 1. So these 2 chromosomes, they will align themselves on the metaphase plate, which is the center of the cell there. And then from there, the spindle fibers are attached to the centromere of the chromosomes. So those are the processes which takes place under Metaphase 2. And then from there, the cell will proceed to Anaphase 2 after the spindle fibers have been attached. So during Anaphase 2, the spindle fibers will contract. Okay, so when they contract, they will split the centromeres, moving the sister chromatids to opposite poles. So the chromosome, the spindle fibers will contract. They'll contract, pulling the sister chromatids to the opposite poles of the cell. As you can see here, the sister chromatids are being pulled to the opposite poles by the spindle fibers which are contracting. So that's what happens under Anaphase 2. Then from there, the cell will proceed to Telophase 2. Now during Telophase 2, the nuclear membrane now here, they will start to reform. Here now the chromosomes have been pulled to the opposite poles, and then in the nuclear membrane will reform around the 4 sets of daughter chromosomes. Okay, and then from there the chromosomes begin to uncoil and decondense. Remember, they were condensed, now here they decondense, and then they uncoil. Then from there, after that, what will happen is that the final result of this division is 4 cells containing haploid sets of chromosomes. Remember, these cells are genetically unidentical. They are unidentical because of the crossover which takes place under Prophase 1. So what will happen is that 4 daughter cells will be formed, meaning the cell here, the cell membrane, the nuclear membrane will start being reformed on the daughters, on the chromosomes which were pulled to the opposite poles of the cell. Okay, and then from there, the cell membrane will start to reconstrict, forming 4 daughter cells. Okay, so this process of reconstriction of the cell is completed by what we call cytokinesis. cytokinesis. So cytokinesis simply separates the two cells into 4 unique haploid cells. So this is the resulting cells which have been formed after metaphase 2. So those are the stages of metaphase 2. In summary, we can say under Prophase 1, the centrosomes are formed, and then from there the spindle fibers emerges, and then from there the nuclear membrane breaks. After that, it will proceed to metaphase 2, where the nucleus or the chromosomes are attached to the spindle fibers, and then from there Anaphase will separate the cells, the chromosomes, and then from there under Telophase they will separate, and the nuclear membrane will be formed around all these cells there. That is under Telophase. And then from there, cytokinesis will separate the cytoplasm, forming 4 daughter unique haploid cells. So those are the stages of metaphase 2. So let's also look at the importance of meiosis. Having known all the stages of meiosis 1 and meiosis 2, what are the importance of meiosis? So meiosis helps to maintain a constant number of chromosomes by reducing the chromosome number into the gametes. So it reduces the chromosome number. It reduces the chromosome number to half, and then as reproduction takes place, we get 1 gamete from the paternal and 1 gamete from the maternal, and form the full set, which is about 46 chromosomes. So by so doing that, it maintains the constant number of chromosomes. Then from there we have it also helps to bring about genetic variations through crossing over and independent assortment of chromosomes. Then after that, it also helps in the formation of gametes and spores for sexual reproduction. So that's all for meiosis 2. Thank you very much for watching. Remember, don't forget to like, subscribe, and leave out your comment in the comment section below.